Good morning, everybody. Today's class is about posterior pituitary, also called as neurohypophysis. Dr. Sandil will be dealing about this. Posterior pituitary, also called as neurohypophysis, is a downgrowth of the pituitary of the hypothalamus. The posterior pituitary is connected to the brain by hypothalamo-hypophyseal tract, also called as neural tract. The hormones are synthesized, and these are all the neuropeptides which are synthesized in the supraoptic and paraventricular nucleus, and they are descended along the posterior pituitary and stored in the granules, the terminal endings of the axon called as herring bodies. In today's class, we'll be dealing about the structure, synthesis, storage, and secretion of anterior pituitary and oxytocin. Oxytocin is a very important hormone. The two neuropeptides are mainly the oxytocin and vasopressin, and they are synthesized in the cell bodies of magnosenal neurons in supraoptic and paraventricular nucleus of hypothalamus. Oxytocin is predominantly synthesized in the paraventricular nucleus, whereas ADH, also called as vasopressin, is synthesized in supraoptic nucleus. The main difference in oxytocin and vasopressin is the third and 9 amino acids are replaced in oxytocin by leucine and isoleucine, whereas in vasopressin it is phenylalanine and arginine. So it's called as arginine vasopressin, as you all see this. The steps involved in the synthesis are, as you all know, the mother molecule for ADH is neurophysin, that is a pre-propresophysin, and for oxytocin it is pro-oxyphysin. Here the leader sequence is cleaved, resulting in formation of the main ADH and oxytocin, which are packaged in the secretory granules of the terminal axons as herring bodies. They are transported along the axons called as axoplasmic flow. The precursor molecules are cleaved in the nerve terminal and are stored in nerve endings. These hormones, which are secreted from the hypothalamus as they are neuropeptides, are called as neurosecretions or neurohormones. These hormones are released into the blood mainly due to electrical activity in the nerve terminal, and which is mediated by calcium dependent exocytosis of hormones from the nerve terminals. So it's called as excitation secretion coupling. Oxytocin, which is the bonding hormone, also called as happy hormone, is a nanopeptide with similar amino acids to ADH, except that the leucine and isoleucine is replaced in the third and eighth position and bound by a disulfide bond. You can see this. The principal site of synthesis of oxytocin is paraventricular nucleus of hypothalamus, and to some extent 1/6 portion is also synthesized in the supraoptic nucleus of hypothalamus. The main actions are on the milk ejection, also called as milk let down reflex, and also results in contraction of uterus. It is mainly mediated by G protein coupled receptors, and oxytocin triggers increase in intracellular calcium levels. The first is the milk ejection reflex, also called as the Ferguson reflex. Here there is stimulation of the touch receptors around the nipple, which is the main receptors, and the stimulus is As the baby suckles the nipple, the afferent information is carried from the mammary gland to the spinal cord via the touch pathway and finally it ends in the paraventricular and supraoptic nucleus of the hypothalamus, and thereby resulting in stimulation of paraventricular nucleus and hypothalamus, which causes action potential to travel down the terminal to the posterior pituitary along the hypothalamo-hypophyseal tract, releasing the oxytocin into the blood. Oxytocin mainly stimulates the myoepithelial cells around the lobules of the breast. We have seen the myoepithelial cells are present even in the extraglobullar apparatus, and thereby, whenever the oxytocin stimulates the myoepithelial cell, it results in ejection of milk, and it's a positive feedback mechanism. More the suckling, more the stimulus, and more the release of oxytocin, which results in a positive feedback mechanism. And this reflex is terminated whenever the baby stops suckling. And this is how the milk ejection reflex is initiated. Whenever the baby suckles, the information is carried along the afferent nerve to the spinal cord, which ascends along the touch pathway, results in release of oxytocin and the paraventricular nucleus, resulting in release of oxytocin, which acts on the myoepithelial cells. The next action is the effect on uterus. In 1906, Dale had used posterior pituitary extract, and which resulted in powerful contraction of the uterus. This process of delivery is mediated by a positive feedback mechanism. What happens is whenever there is parturition, the endometrial sensitivity to oxytocin is increased by estrogen, and thereby it enhances the increase in oxytocin receptors and decreased by progesterone. As you all know, the action of oxytocin or the half-life of oxytocin is only 3 to 5 minutes, and you can see the expulsion of fetus is called as descent or is called as parturition. What are the steps involved? What is the mechanism of parturition? Whenever there is a descent of fetus, there is dilation of the cervix, which results in stretch of the cervix and thereby causing stimulation of stretch receptor, which increases the activity of the afferent nerve, which in turn stimulates the paraventricular nuclei, thereby releasing oxytocin and causing contraction of uterus. More the descent, there is more dilatation and more oxytocin is released, and thereby causing expulsion of fetus. And the expulsion of fetus is called as parturition. This is a diagram which depicts how parturition is initiated. Whenever the baby head stretches the cervix, it causes fundic contraction, and this fundic contraction pushes the baby down and thereby stretches the cervix, and this cycle repeats over and over, causing expulsion of fetus. Both these milk ejection reflex and parturition reflex are called as neuroendocrine reflex because it's a reflex in which the nervous system and endocrine system interact in bringing about the reflex. You can see in a normal reflex arc, which is an automatic response to a stimulus, there is a receptor and afferent with a integrating center in the CNS, and eventually the end organ is a muscle or a nerve and results in a neurotransmitter release. Whereas in neuroendocrine reflex, there is a receptor, an afferent arm with an integrating center in the hypothalamus, resulting in release of oxytocin and which acts on the end organ, mainly the breast and uterus. The other actions are during menstruation, it results in luteolysis, that is breakdown of the corpus luteum, and contraction of the vas deferens, and also it results in fallopian tube motility. The clinical relevance is it helps in induction of labor and postpartum hemorrhage. Whenever the labor is delayed, it results in induction of labor. The synthesized form of oxytocin is also called as syntocinon, whereas the second clinical relevance is during the active stage of third stage of labor, we give 10 units of oxytocin to prevent postpartum or antepartum hemorrhage. The next neurohormone which we'll be dealing is the arginine vasopressin, also called as antidiuretic hormone. This hormone prevents diuresis and causes, not only prevents diuresis, it also causes very important action on blood vessels by initiating vasoconstriction. It's predominantly secreted by supraoptic nuclei, and the biological half-life is around 18 minutes. It is degraded in the liver and kidneys. It's a nanopeptide with phenylalanine and arginine in third and ninth position. The actions of vasopressins are mainly maintenance of the extracellular fluid volume and osmolarity. It also acts on the distal convoluted tubule and collecting tubule, thereby increases the water permeability. 90% of the water reabsorption takes place in the proximal convoluted tubule, only 10% of reabsorption takes place in the DCT and collecting tubule, and thereby results in reabsorption of water, which in turn maintains the volume, extracellular fluid volume. As you all know, the maintenance of extracellular fluid volume is more important than maintenance of osmolarity. It is a potent vasoconstrictor in pharmacological doses, not in physiological concentration. However, it is secreted or released during emergency like hemorrhage, and thereby it's a protective phenomenon causing vasoconstriction. It is mediated by V1A receptors. The receptors mainly which the ADH acts are the V1A, V1B and V2. V1A is predominantly in the vascular smooth muscle, whereas V1B is in the anterior pituitary to cause release of ACTH from the anterior pituitary. The V2 receptors are acting on the distal convoluted and the collecting tubule, mainly in the principal cells where the water channels or the aqueous channels called as aquaporins are inserted, resulting in increased water permeability and thereby water is reabsorbed. The actions are water reabsorption, vasoconstriction, a regulation of extracellular fluid volume and blood pressure, and also it stimulates the release of corticotropin releasing hormone from hypothalamus, and also it facilitates memory and causes breakdown of glycogen, resulting in glycogenolysis. What is the mechanism of action of ADH? ADH binds to the V2 receptors in the distal convoluted tubule and causes, what does it cause? It initiates the conversion of ATP to cyclic AMP, mainly by activation of protein kinase. Whenever the protein kinase increases, there is increase in cyclic AMP and causing. So ADH binds to specific plasma receptors on the basal portion of the cell and activates adenyl cyclase. This causes activation of protein kinase on the luminal side of the cell. The activated protein kinase causes insertion of protein water channels called as aquaporins, which predominantly aquaporin 2. Aquaporin 1 is in the proximal convoluted tubule, thereby resulting in water to move rapidly from the tubular lumen into the cells through these aquaporins. This is mainly in the principal cells of the distal convoluted tubule. It's a gene mediated pathway, it's a genomic pathway where there is gene transcription and increased aquaporin 2 formation. So it is regulated by mainly the osmolarity and the changes in extracellular fluid volume. There are receptors for osmoregulation. There are receptors which are present in the anterior hypothalamus called as osmoreceptors. They are stimulated by increase in osmotic pressure of the body fluids. When they lose water, the receptors are stimulated, which in turn stimulate the neurons which secretes ADH, so thereby resulting in more water to be reabsorbed in the kidney and the osmotic pressure of the blood is brought back to normal. On the other hand, when osmotic pressure falls, ADH secretion also decreases and thereby water is excreted through urine. The second mechanism by which the ADH is regulated is by changes in extracellular fluid volume, also called as the volume effects. Here, vasopressin is secreted when the extracellular fluid volume is low, and it is decreased when the extracellular fluid volume is high. It is mainly mediated by stretch receptors. There are two types of stretch receptors. One is the low pressure and the high pressure receptors. The low pressure receptors are present in the great veins and atria, and these receptors are mediated of volume effects on the vasopressin. Hypovolemia and hypotension release large amounts of vasopressin. ADH is increased by increase in osmotic pressure, decrease in extracellular fluid volume, and also due to pain and exercise, whereas it is decreased due to decrease in osmotic pressure and extracellular fluid volume, and also by action of alcohol and catecholamines. So this is how it can act. Whenever there is diarrhea and vomiting, there is loss of water, it decreases the extracellular fluid volume. The low pressure receptors are stimulated, which enhances the vagal activity, that is, it decreases the vagal activity, which in turn causes the ADH to be secreted, and there is water reabsorption, and thereby the extracellular fluid volume is restored. Whenever there is increased plasma osmolarity, the osmoreceptors are stimulated in the hypothalamus, which enhances ADH to be secreted and resulting in increased plasma ADH, which in turn causes water reabsorption, and again the extracellular fluid volume is restored. Whenever the BP is decreased, the high pressure receptors are stimulated, which in turn increases the activity in the nucleus tractus solitarius and medulla, and thereby resulting in ADH secretion, which enhances water reabsorption and causes restoration of extracellular fluid volume. The clinical relevance is whenever there is deficiency of ADH, it results in diabetes insipidus. We came across diabetes mellitus, which is due to deficiency of insulin, whereas when there is deficiency of ADH, it results in diabetes insipidus. There are two types, neurogenic and nephrogenic. When there is absolute deficiency of ADH, it results in neurogenic type of diabetes insipidus. When there is a mutation in the V2 receptors, it results in nephrogenic type. And there is another disorder called a syndrome of inappropriate hypersecretion of ADH secretion. Diabetes insipidus is characterized by polyuria and polydipsia, which is nothing but passage of large amounts of dilute urine and drinking large amounts of water. Dehydration may occur in severe cases leading to hypotension. Here there is decreased ADH secretion. The central type of is also called as neurogenic, where there is decreased synthesis or secretion which can be due to inherited or acquired. Inherited is mainly genetic, it is mainly the autosomal dominant Wolfram syndrome, whereas if there is a tumors in the supraoptic, it is mainly by the acquired. Nephrogenic is failure of ADH sensitive receptors, that is the V2 receptors in the nephrons. There is diuresis, which eventually causes the urine to be dilute and hypotonic. Syndrome of inappropriate ADH secretion is characterized by excessive secretion of ADH, which is resulting in renal excretion of sodium, causing hyponatremia, increase in the blood volume, and hypoosmolality accompanied by neurological signs like irritability, confusion, muscular weakness, and seizures. In certain cerebral disease and lung diseases like lung tumors, the impulses from the volume receptors do not reach the hypothalamus to inhibit ADH secretion. The body osmolarity will be low, but ADH secretion will be high. The extracellular sodium level will be very much decreased and extracellular fluid volume will be high. There will be features of water intoxication. To summarize, oxytocin is secreted by the paraventricular nucleus and supraoptic causes release of ADH. So ADH is mainly causing water reabsorption through the B2 receptors by water channels called as aquaporins. Oxytocin acts as action on breast by causing milk ejection reflex, and parturition, which is expulsion of fetus. These both reflexes are neuroendocrine reflexes. Thank you.